hi, hello. So good evening, this is Krishnaveni, your biology faculty, and we are on this journey talking about the story of life on earth, that's biology, right? Okay, so good evening. So good evening, Priyanka. So it's nice to see you guys on time. So okay, so please don't forget to like, share and subscribe. So do it now, because your small like, share and subscription can at least open the door for people who are not aware of this opportunity. So there are a lot of people who cannot afford for a paid class. So at least for them, they will actually enjoy the benefits of this. Okay, so there are a lot of NEET aspirants today in India. At least at the time of lockdown, so many people are suffering. So your small like and subscription can enable people to understand that the reach is wide. Okay, so they are open to a lot of opportunities out before them. Fine. So please do like, share and subscribe. So please tell your friends that there's something going on like this called the NEET class in English medium. Okay. So we have all the 3 subjects going on for both the standards 11th and 12th. So please let your friends know. So share as much as you can. Fine. Okay. So thank you, Priyanka. Good evening, Harini. Okay. So today it's Wednesday. So we'll be talking about class 12 portion. That is, we'll be talking about the world of genetics. So Monday, Wednesday and Friday, I'll be talking about class 12 syllabus. And the rest of the remaining 3 days of the week, I'll be talking about class 11. So please stay tuned. I promise to make boring lectures interesting as well. Yes, good evening. That's good. So it's good to see all of you excited. So even I am excited. Fine. So let's go forward and see what we have today. So let's start this evening with a quote. So I know you're already tired after watching your physics and a chemistry lecture. So this is the last lecture, that is biology. So let's make it short, because we'll be talking about the central dogma of life, the continuation of the previous lecture. But still, your human brain cannot concentrate for more time, right? It also has a threshold level. It also has a saturation limit. So we are talking about biology, so we should know how your brain reacts, right? So let's make it interesting and let's make it short, crisp and sweet. Fine? Fine. Okay. So the quote for today, it says, we swim in a sea of science. So just like the fish which takes water for granted, we take science for granted. But without science, we would all die. So without science, there's no life, right? So at least for NEET aspirants, without physics, chemistry, bio, there's no life. But talking about reality also, so without science, there's no life. So science is the ultimate thing. So we're all swimming together in the ocean of science, trying to discover new things, trying to enjoy the discoveries that was already done. But still, we are craving for more, we are thirsty for more, right? But we still don't know we are enjoying the happiness of science, we are enjoying the fruit of science. So science, without science, there's nothing. So talking with respect to biology, so like I told you in the first class, so biology is the natural science of life. So we are talking about biology here. So biology is easily relatable, because what's right now happening in your body, so that makes you so fascinating, right? Yes. So if not now, very soon you will do. Okay. So hi, Sheikh. Fine, you're a CP student. Okay. So going forward, so we were here talking about the world of genetics. So last class I was telling you that the best gift that you can expect from your father and mother is what? Okay, sorry. So the best gift that you can expect from your parents, that is from your father and mother, is 2 sets of 3 million DNA. So that is your genetic information, and this gift is so fragile, so important and so fundamental for you to perform today. So today if you're walking, if you're looking around, if you're talking, and it's because of those 2 sets of triple DNA codes that you have, right? So we were talking about that. So we were so excited that we started with the world of genetics. So I told you about the classification of genetics as well. So currently we are talking about the molecular genetics, right? So genetics is as such interesting. So we were talking why genetics is interesting, right? So today we are in the era of evolution. We are in the era of genetic evolution, where everything is becoming genetic. You can make an artificial baby. You can have an artificial hand. So everything is possible, right? So you have the power of God. You can create life. So that's only possible because of genetics, right? Do you know you can create life? So just culture. So a normal skin cell can be grown. So anything is possible. So you have the power of a Promethean God. But this ability or this responsibility to create life has a lot to be understood. So you have a lot of ethnic responsibilities. You have a lot of limitations. You have a lot of challenges. So everything can be blamed on genes. So today's newspaper, when you look into the newspaper, all that you see is about coronavirus, about schools opening in South, and a lot of other things, right? But let me tell you, it's a promise actually. So write down 30 years or another 25 years, you will only talk about genetics, we will only talk about genes, gene therapy, etc., etc. So it will be all in the genetic world. So recently the Nobel Prize was also given for the CRISPR genome editing, right? So everything is genes. You can do amazing things with genes because they are the basis of life. So with that, we started our lecture. So our first lecture is over. This is our second lecture. So going forward, okay, so we were talking here, right? Why genetics is interesting. If you remember this slide, so you can blame everything to your genes. So why genetics has to fascinate you? So you can replicate faithfully. So what is being passed on from one generation to the other? So what is being produced? What is happening when you're going to reproduce? So this dog, the mother dog, the puppy resembles your mother dog, right? So you resemble your mother, right? So all this happens because of genetic. So the fundamental information that's being passed on, right? So that is why genetics is fascinating. So let me tell you another news. So when we go to the next slide, you will actually understand why genetics is fascinating. Okay, so I have a happy news again. So there's something called as a happiness gene that is discovered. Can you imagine something as simple and as silly that a happiness gene is present inside your body? So if you're happy, it's because your gene is active. If you're sad, it's because your gene is off. Can you believe it? Trust me, it's true. If you're lazy, that's because you have more of lazy genes. If you're active, that's because you have less of lazy genes. Okay? So everything can be attributed to genes. So there's one amazing book on genes and it is called Selfish Genes by Richard Dawkins. So if you're really fascinated about genetics, so please go ahead and read that book. So it takes actually 3-4 readings to understand what he's trying to contemplate. So his level of genetic understanding is brilliantly amazing. Okay, so it takes 3-4 readings to understand what is he actually trying to say. Okay, so then you can enjoy the essence of the book. Okay, so there's something called as happiness gene that is discovered. So this gene has a strong influence on how your life is. So whether you're happy, so when you look at some people, they're always happy, right? They're always smiling, right? So that makes you happy. So they spread happiness because that has a strong influence on other people as well. So it is all because of your happiness genes. Fine. So what is actually that gene? So that gene is called as 5-HTT. So that is a particular name of the gene. So it is a serotonin transporter. So if you have time, just go Google it and find what is a serotonin transporter. So basically serotonin is a neurotransmitter and the equal distribution of serotonin ensures you are happy. Okay, when your distribution of serotonin is not uniform, it's not even, you're not happy always. Okay, so now just look at yourself in the mirror and tell me if you're happy or sad. So if you're happy, it means that this gene is perfect. If you're sad, it means that you have a defect in this gene. Okay, fine. Okay, so now you find and give me the answer whether you're happy or sad. Okay, so going back to today's talk. So where were we? We were talking about the central dogma of life. So what is central dogma? So dogma is something that is inevitably proved. Okay, so it's a set of principles that have been proved, that have been validated. Okay, so that is called as dogma. So further, we were talking about replication. So we were done with replication, right? So let me take my pen. So we were done with replication the other class. So that was the first class. So today we'll be talking about transcription. Sorry. So just a second. Okay. So today we'll be talking about transcription. Fine. Okay, just give me a minute. Okay, so just there's some technical issue. Fine. So just let me explain it to you here. So the central dogma of life. So you have replication, you have transcription, and you have translation, right? Hold on, I'll come back. Okay, fine. So sorry for the interruption. So we were talking about the central dogma of life, we were talking about replication, we were talking about transcription and translation, right? So quickly recapitulating, so your central dogma, so this GIF summarizes your central dogma. So you have your information in the form of DNA, right? So from DNA, it is copied on to RNA. So it's basically mRNA and then finally to proteins, right? So that is your central dogma of life. So going forward, okay, now let me quickly recapitulate what we were seeing last class. So last class we were talking about DNA. So DNA is your genetic material. So DNA stands for deoxyribonucleic acid. So what is your DNA made of? So your DNA is a polymer of nucleotides, right? So what is a nucleotide made of? A nucleotide is made up of a nucleoside plus a phosphate base. So I'm quickly recapitulating to you. So you have three components of a nucleotide. So you have a nitrogenous base, you have a sugar molecule, you have a phosphate, right? So your nitrogenous bases are your purines and your pyrimidines, fine. So your purines are adenine and guanine. So your pyrimidines are the remaining C, cytosine, thymine and uracil. So these three are your purines and these three are your, this is your pyrimidines and this is your purines. So the difference between them is this has two aromatic rings and this has a single ring. So what is the sugar in case of your DNA? So it is the deoxyribose sugar. And finally you have your phosphate group, okay? So when your nitrogen base combines with a sugar, it is called as a nucleoside. So do you remember? We were talking about this in the last class. So good afternoon, Deepak. Okay, so Deepak is asking when the class started. Okay, this class started at 4. Okay, so today we were pretty late by 10 minutes. Okay, so good afternoon, Himanshu. Okay, so the sugar is deoxyribose sugar. Okay, so this is the nucleoside. So when you have your nitrogenous base which is bound to the sugar molecule, it is called as nucleoside. And when a nucleoside binds to a phosphate group, it is called as a nucleotide. So finally, that is the nucleotide that we were talking about. So how is your DNA formed? So your DNA is like this, right? So your bases are like this. So you have ATGC. So what was the hallmark of DNA? That is a complementary base pairing. So this pairs with T. So this was one of the hallmarks of your DNA, right? So you have complementary base pairing. The second hallmark was 5' to 3', 3' to 5'. It has anti-parallel polarity, right? So this is what we were talking about in last class when I was giving you the introduction to DNA. Fine. So last class I didn't tell you about the types of DNA. So there are 3 types of DNA. So you have your B DNA, A DNA and Z DNA. Fine. So B DNA is your normal canonical DNA that we are talking about. So it was given to us by your Watson and Crick. Okay. So that is your normal B DNA. So in B DNA you have 10 base pairs per turn. So I mean to say your DNA is a double helix. So per turn you have 10 base pairs. Fine. So A DNA has 11 base pairs per turn and it is always used in solutions which is devoid of water. So basically it is a structural variant of your B DNA. Fine. Okay. And your Z DNA is a bit slender and elongated. So it has 12 base pairs. Fine. So today's research suggests that there are stretches of Z DNA present in eukaryotes. That is, you and I have stretches of Z DNA. So that is the recent discovery that is being found. Fine. Is this okay? So shall we move forward? So this is a quick recapitulation. So moving forward, now we'll talk about replication in short. So what was the first thing we spoke about replication? Yes. So anti-parallel is one of the hallmarks, right, Priyanka? So your B DNA is the normal DNA. So the DNA that you and I have, the canonical one, is your B DNA. So that was given to us by Watson and Crick. So it is the Watson and Crick form of DNA. Okay, fine. So talking about replication in short, so we were talking about all these terms. So let me quickly recapitulate. So these were the terms that we were talking about, and this is how your DNA unwinds, right? This is how your DNA unwinds. So that is our first step, because your DNA has a long stretch. So the entire DNA will not unwind simultaneously, but it will unwind at a particular place, and that is called as your replication bubble. Fine. And your replication will initiate at a particular point, that is called as origin. Okay. And you have a player. So the most important player here is your DNA polymerase. So your DNA polymerase helps in polymerizing your new strand of DNA. Fine, okay. And the last one is Okazaki fragments. So what is Okazaki fragments? So this is how actually your replication occurs. So if you are not quite clear with this, so let me quickly recapitulate it for you. Let's go to the next slide. So what happens? You have your DNA, right? So this is your 5' end, this is your 3' end, this is your 3' end, this is your 5' end, right? So it has anti-parallel polarity. So this is your DNA. So we are going to look into how your DNA is going to replicate. So you have your strands being unwind. So unwinding of the DNA strand is called as, is done by helicase, right? Yes, it is DNA dependent DNA polymerase, right? So now let me label 3' to 5', sorry, okay, 5' to 3', 3' to 5'. So who's the first player here? DNA polymerase. So DNA dependent DNA polymerase. Why it is called as DNA dependent DNA polymerase? It's because your template is your DNA here, right? So that is why it is called as DNA dependent DNA polymerase. So your DNA polymerase has a starting trouble that I was talking about. It cannot initiate synthesis, but it can initiate synthesis from a primer. So one more important point is that your DNA polymerase can only synthesize in the 5' to 3' direction. Fine. So here the synthesis is continuous. So this is how you have your synthesis, and this is the direction of your replication fork. So your DNA will begin to unwind in this direction. Fine. Is this clear? But look at the other strand. Here it has to happen in the reverse direction. So it will happen in short fragments. So these fragments are called as Okazaki fragments. Fine. So later, though these fragments are discontinuously synthesized, they will be later joined by your DNA ligase. Fine. So this is what we were talking about in the last class. So basically we were talking about replication more in detail. So this is a quick summary of your last class. For those who are new, this is good. Yes, so this is called as a leading strand. This is called as a lagging strand because it is also the discontinuous strand. So this is synthesized in a discontinuous way, right? That's why. Fine. So going forward, now we actually started with transcription, but we didn't go deep into transcription last class because we were already running short of time. So transcription, so what do you think is transcription? So transcription is copying. So now your DNA is ready. So your DNA has replicated. Your DNA is perfectly ready. It has produced faithful copies of itself. Now it has to pass on the information or copy the information onto an mRNA. So that process is called as transcription. Yes. So copying genetic information from one strand of DNA into RNA is called as transcription. So what governs this process, or what is the driving force of this process, is your principle of complementary. So you have complementary base pairing. So that is the reason that the process of transcription runs properly. But this was the question that we were discussing last class. Why not both the strands of DNA are copied? So in case of replication, both the strands of DNA were replicated simultaneously, though continuously and discontinuously, but it was done together, right? But in case of transcription, not both the strands of DNA are copied. So why do you think is the case? So we have discussed it in the last class. So the first reason is that if both the strands are going to be copied, so you'll have two set of proteins that are being produced. Okay, so those two set of proteins will again complicate your genetic machinery. Another logical reason is your DNA already has complementary base pairing, right? For suppose, let's take a strand of DNA. So this is one strand, this is another strand of DNA, fine, okay. So here this information is copied on to mRNA 1, this information is copied on to mRNA 2. So these two strands are complementary. If the information is copied on to mRNA, won't these two be complementary? Obviously it will be complementary, right? Now if these two are complementary, it will come and form a double-stranded DNA. Yes, right. Okay, so when it forms a double-stranded DNA, your last process of translation will not happen. Fine, okay. So that is the reason not both the strands are copied here. Okay, so we were talking about the transcription unit. So your transcription unit has three regions. So what are they? So one is called as promoter, the middle one is called as your structural gene, and finally it is called as your terminator. Fine. So what was the figure that we were talking about? So basically this. So this is what we were talking about, if you remember. So transcription is processed by what is called as a transcription unit. So that consists of 3 members: your promoter, your structural gene, and your terminator. Fine? Is this clear? Okay. So going forward, so we'll talk more about transcription. Okay. So this is how it happens. So you have your coding region. So here you have your terminator region. So here you have your promoter region. Fine? Okay. So which enzyme catalyzes DNA replication during transcription? So what question is this? So replication is a separate process, transcription is a separate process. So DNA replication is catalyzed by your DNA dependent DNA polymerase. Fine. In case of transcription, the player is your RNA polymerase, DNA dependent RNA polymerase. So your replication transcription is not a continuous process in case of eukaryotes. Fine. Okay, so Priyanka Singh, are you getting my point? So your question says which enzyme catalyzes DNA replication during transcription. So these two are not a combined process, it's a totally different process. Fine. So DNA replication, when it happens during the S phase of your cell cycle, right? Fine. So this is how it happens. So this is transcription. So basically what I am trying to tell you is you have your DNA, right? So here the main player is your RNA polymerase. Okay. So RNA polymerase again has a rule that it will synthesize information, or it will work only in one direction. It will work only in the 3' to 5' direction, meaning this direction. Okay. So only in this direction your RNA polymerase will work. Fine. So thus your strand, which has 3' to 5' complementarity, will act as a template strand. Fine. The other strand, though it does not code for anything, it is called as coding strand. Because the rule of transcription is that only one strand is copied, right? So the strand with 3' to 5' is only copied, okay? And that strand acts as a template here. Okay, fine. The other strand acts as a coding strand. So it has a weird name, though it does not do anything. It's given the preference of, it is given the honor of calling the coding strand. So your coding strand is also known as the sense strand, and your template strand or the non-coding strand is also known as the anti-sense strand. Fine? Is this much clear? So this is in layman terms how transcription works. So finally your polymerase will produce an mRNA here, right? But how transcription happens in eukaryotes and prokaryotes is pretty different. So they are not the same. So the process of transcription and transcription in eukaryotes and prokaryotes is pretty different, because in prokaryotes your genome is not very complicated, but in eukaryotes the genome is pretty complicated. Fine. So going forward, okay, so this is what we were talking about in the last class. If you remember this slide, so this is exactly where we stopped. So how do you define a gene? So gene is the functional unit of inheritance. Also we were also defining gene as a sequence on a nucleotide or a polypeptide which can code for a protein. So that is also how gene is defined. Now we have another synonymous or analog for gene. It is called a cistron. So cistron is the sequence which codes for a polypeptide. Fine. So your cistron is also equivalent to your gene. So there are two types of cistrons, monocistronic and polycistronic. So mono, as the term says, it stands for one. Poly means many. Fine. So that is monocistronic and polycistronic. Yes, monocistronic is for eukaryotes. So your bacteria, though the genome is not very complex, so all the structural genes, all the important genes of your bacteria, they are clustered together. So that is why it is called as polycistronic in case of prokaryotes. So remember P and P. So prokaryotes have polycistronic gene, okay, and your eukaryotes have monocistronic gene. Is that fine? Is that understood? Okay. So your prokaryote is pretty simple. So this is your prokaryotic genome. So you have your 5', 3'. So you have your 3', 5'. So you have your promoter region. Here you have your terminator region. And then the information is copied. It's pretty simple. But in case of your eukaryotes, you have your promoter, you have your terminator. In between, there are complexities. For example, I will shade. So this is called as exon. Let's say the shaded ones are called as exon. and the middle ones are called as introns. So just as you can see it here. So exons are the ones that actually code for a protein, and introns do not code for protein. So the introns are basically your disturbing sequences or your intertwining sequences. Fine. So now for information to be copied, this intron has to be removed. Why do you need unnecessary information? Okay, so from which strand coding of mRNA takes place? So your mRNA, for your transcription of mRNA, it happens in your 5' to the 3' direction. So the strand which has 3' to 5' polarity, it acts as a template strand. So from that strand, so basically from the lower strand, the information is copied. So now you have to remove the introns from here. So the initial pre-mRNA will have introns and exons. Okay, so this is not in case of your DNA. So once you have your mRNA produced, so actually I drew it in DNA, it is not in case of DNA. So this is, we are talking about DNA. Wait, let me get it right. So let me redraw it for you. So in case of prokaryotes, you have your genome like this. It directly produces an mRNA, a single stranded mRNA. So it is pretty simple. So all your genes are clustered together, and that is called as polycistronic. Fine, no confusion. But in case of your eukaryotes, okay, so eukaryotes also have double stranded and you have an mRNA produced. But how is the mRNA? So the mRNA has intertwining sequences in between. So this is your exon, which actually codes for a protein, and these are introns. They are just disturbing sequences. So earlier these introns were called as junk DNAs because they do not code for a protein. Then why are they present? So thus introns were initially called as junk DNA. So our body has a mechanism by which it can remove the introns and only contain the exons. So that is why the transcription in eukaryotes is a bit complex. Okay, since you have one gene at a particular interval, it is called monocistronic. Fine, so it is not clustered together. But today we know that these introns are not junk DNA, but they actually play a role in the process of transcription. So we don't know exactly how, but studies are going on as to how these play an important role in transcription. Fine. So going forward and looking into the transcription of your bacteria. So bacterial transcription is polycistronic, so that is understood. So there are three basic steps in your bacterial translation. So what are the steps? So first it has initiation, then it has elongation, and finally termination. So as the name says, first there is someone to initiate transcription. So here in case of a central dogma, all players have a starting trouble. So someone has to come and initiate the process. Then it will elongate, meaning your mRNA is being produced. Once your mRNA is produced, it will terminate completely. Your process of transcription will end. Fine? Okay. So here you have RNA polymerase. So this is DNA dependent RNA polymerase because your template is DNA. Fine. So look here, you have what is called a sigma factor. So when your sigma factor comes and binds, okay, yes, your rho factor, yes, you're right, Priyanka. So it comes and binds to the promoter region. So okay, now your promoter understands, okay, now we have to start the process of transcription. So the process of transcription proceeds. Okay, so you have elongation. So as your RNA polymerase moves forward, so that is called as elongation. You have your mRNA being produced. Finally, when it is done, your RNA polymerase will detach from the DNA. So that is called as termination. So you have three simple processes: initiation, elongation, and termination. Fine? Is this done? Okay. Now we are going into eukaryotes. So in eukaryotes, it is pretty complex because we have to splice out what is called as introns, right? So we only need exons, that is the ones which are coding, and not the ones which are non-coding. Fine. So let's see how it happens in case of eukaryotes. So in eukaryotes, once the pre-mRNA is produced. So what is the pre-mRNA? From your DNA, so this is your double-stranded DNA. From your double-stranded DNA, you have mRNA being produced, right? So this is called as pre-mRNA because it contains both your exons and introns. Now our primary aim, our objective is to chop off that introns and only have exons because exons can code for proteins, right? So that is our agenda. So this is called a pre-mRNA. How to chop off that introns? So that is what we are going to see. So the first step that happens is capping. Okay, so capping, so this pre-mRNA is being capped. So it is capped at the 5' end. It is capped at this end. So the cap is called as methyl guanosine triphosphate. So basically it is capped because it is an immature mRNA. It can be prone to degradation because your body has a machinery where you have sentinels or you have guards which will look out for proteins which are of no use. So the moment it looks at your pre-mRNA, it can directly attack your pre-mRNA. Thus, to protect it from degradation, it first wears a cap. So that cap is called as methyl guanosine triphosphate. Fine. So the next process is called as splicing. So splicing is the process where you actually chop off your introns. You only take out your exons and you throw out your introns. Fine. That is the process of splicing. And then the third process is tailing. So first it's capped. Now you have removed all your introns. So it becomes like this. So it still wears the cap. Finally, once the introns are removed, you will have tails. So they are called as poly A tails. Again, this tailing happens to protect the mRNA from further degradation. Fine. So now I have a picture. So this is your immature, this is your double-stranded DNA, right? So your RNA polymerase is acting on it. So you have your mRNA coming out. So this mRNA is your pre-mRNA and it is pretty highly unstable. Okay. So it immediately wears a cap to prevent itself from degradation, right? So once it has been capped, now your introns are being chopped off. So they are being spliced out. So that is the process of splicing. Once it has been spliced off, once you have your mature mRNA, it is quickly tailed. So you add polyadenylate residues to it. So actually this addition of polyadenylate residue acts as a signal for some process inside your body. Okay. So this is the process of transcription in eukaryotes. Fine. So going forward, we are entering into translation. So we are done with the two steps of central dogma. We are done with what is replication and what is transcription. So can you imagine if your body lags behind in any process? If your body says today I will replicate, tomorrow I will transcribe, after a month I will translate. So what will happen? Anyways, we are already very slow, right? So imagine our lag then. So every process has to happen efficiently. It has to happen correctly, because if something goes wrong, the entire mechanism of your body goes wrong. So remember, all your body parts are coordinated. For example, if you diagnose a cancer at a stage 4, okay, at the final stage of cancer. So be it any cancer. For example, if you have, say if you have breast cancer, so at final stage, it's not only that region that is affected. So your brain is affected, your tumor is affected, everything is affected. Say if you have blood cancer, not only your blood carrying vessels are affected, your brain is affected, your ears are infected, your kidney failure happens, because everything is coordinated, right? So they can just directly metabolize or metastasize. So that is the serious problem, because all your cells are coordinated. So we are happy in one way because your cell is coordinated, you and I are normal. But when it comes to the opposite side or the negative side, your coordination creates the biggest problem. So that is the reason why the healthcare industry is lagging behind, because we can grow cells on a plate, but we cannot coordinate them, right? So artificially, when I grow cells, I cannot connect them with the brain or with your endocrine gland. So your endocrine glands also play a very essential role, right? So that is the problem we are facing in the healthcare industry today. So now we are going to talk about translation. So this is the final process of central dogma of life, and your ultimate aim of your protein, of your cell, is to produce proteins. Okay. So translation is basically copying information or decoding or translating the information of your mRNA into proteins. So here your mRNA is a polymer of nucleotides and your protein is a polymer of amino acids. So there are two different zones. So DNA, mRNA, there is some match because both are nucleotides. So both are of the same origin, except that the sugar base is different. Fine. But when you talk about translation, so what actually happens is, you are transferring or translating information from a nucleotide to an amino acid. So both does not have any connection. So thus the problem of translation, or the challenge was translation, to find out how cells actually translate proteins, was very, very tough. So it took years. So for a long time we were actually stuck with only replication and transcription. We didn't know how translation happens. So to decode this, to identify this, you had scientists from all over the fields. So you had a physicist, you had a chemist, you had a genetist, you had a biochemist. All of them came together to analyze what is happening. So here I will show you what they found out, and that was the breathtaking discovery of your genetics. That was the breathtaking discovery of your central dogma of life. So this is the problem. There is no complementarity between nucleotides and amino acids, because your nucleotides here are your mRNA, your amino acids here are your proteins, right? So there was no complementary. So in transcription it was very easy, right? Because you had the principle of complementary that was acting, so it was easy for you to quickly copy the information. But in case of translation, it was not possible. So thus the major event, the phenomenal, the groundbreaking discovery was done, genetic code. So we should be proud because an Indian was also involved in cracking the genetic code. So he is called as Hargobind Khurana. So you have fellowships called as a Khurana fellowship. So all those interested in genetic research, so after your degree you can actually go for this Khurana fellowship. So it's still valid, okay? So what did they actually do? So what was this logic of genetic code? So they found out that your mRNA is made, right? So your mature mRNA is made. Now, so your mRNA basically is in the form of nucleotides, right? So it has AUGC. It does not have T, so that is clear. So that is the difference between DNA and RNA. So your DNA has thymine, your RNA has uracil instead of thymine, right? Fine. Now, they found out that there are how many bases? 4 bases. That is AUGC. But you have 20 amino acids in total. So they took out permutation and combination as to how 4 letters can code for 20 amino acids. So it was a basic permutation and combination. So when they did 4 square, so 4 square is 16. So which means it cannot code for 20 amino acids, right? So thus the permutation commutation was selected. So 4 part 3, so it was understood that we have 64 codons. Okay, so 64 codons is what is present in your body right now, and each one codes for a specific amino acids. Fine. So the person who was majorly involved in this was George Gamow. Fine. So what is this genetic code? So we have 4 codes. So you write the codes like this. Okay, so your first base, second base, and then you write it like this. So then you multiply and write. So this is how your genetic code was made. AG. So U, here also U. So the first letter here is U. So this is how you write the genetic code. Fine. So each code now codes for an amino acid. So these amino acids are the building blocks of your proteins, and thus what is called a central dogma of life was made successful. I was just explaining, but I have the code in the next page. So you should really be astounded as to how they found out this. So we are not able to crack a simple NEET questions. So though we have already prepared, but imagine this was created. So you and I didn't create. We are just trying to explore. We are just trying to decode what is happening in our body. And this was one of the phenomenal discoveries that was done. So these are the bases. So you have 4 bases in your RNA. So your first base, second base, and the third base. So you multiply just like you do in your Punnett square. So you multiply and write. So each code, so your triplet code, it codes for an amino acids. So totally there are 64 codons. You call it as codons, and this is called as genetic code. So now what I am trying to tell you is, you have your mRNA. So your mRNA has sequences like A, T, U, G, right? Now let me read this code, A, T, U. So quickly find on, okay, so we don't have T here, I am sorry. So let's take this sequence. So now first let's take the first 3 words, A, C, U. So tell me where A, C, U comes here, right? Now your translation machinery will identify this as threonine, and thus your threonine amino acid will be produced. Fine. So that is how it happens, the process of translation happens, and that is how your genetic code was deciphered. Okay. Yes, there are some sites called A site, P site, E site in translation process. It occurs in your ribosomes. We'll talk about those sites. So if you go back to my previous slide. So here you could have seen, okay, so it was the previous slide. So let me show you the figure as to what actually happens in translation. See, so this is your ribosome. So you have your 3-letter code, right? So this is your P site, this is your A site. So your E site is the one that is bringing in the amino acids. Fine. So see, it starts building the amino acid. So you have the proteins being built. So this is called your ribosome, right? So we have been talking about the cell yesterday. So this is your ribosome, the bigger subunit, the smaller subunit. So this is your mRNA. So your mRNA contains codons. So we read it in triplets. So it is called as a triplet codon. So for example, AUG codes for methionine. So methionine is added. Fine. So this is your tRNA that is performing the role. So this is P side, this is A side. Fine. So now you got a gist of what is happening, right? Okay. So now I'm going forward. So we are done with this. Okay. So I showed you the genetic code, right? Now there are some salient features or something unique about the genetic code. So the genetic code discovery itself was very unique and very, what to say? Very critical actually. So today we are talking about Mendelian disorders. We talk about sickle cell anemia. So what do you think is actually sickle cell anemia? One of the amino acid is misread. So what is being misread? So one codon. So for example, if your codon is 3-letter GUG, if this changes to GUC, then your amino acid changes, then your phenotype changes. So that is what is happening in Mendelian disorders. Okay, so that is called as genetic disorder. So your body is not able to read the code properly. So this was one of the major or the astounding discoveries, but it has some salient features to it. Okay, so let's look at the salient features. So each code is a triplet. So you read it in terms of 3. Okay, so you have totally 61 codons which codes for amino acids, and the remaining 3 codons are stop codons. So just like in the process of transcription, you had what is called as a terminator. So how do you know till when your ribosomes or your tRNA has to read the code? So till it encounters a stop codon. For example, your mRNA is long. It has so many codes here. So finally, when it has a stop codon, it will stop the process of translation. So that is the role of your stop codon. So together you have 64 codons, but 61 codons code for amino acids, and the remaining 3 they code for your stop codons. Fine. So that is the first feature of your genetic code. The second one, some amino acids are coded by more than 1 codon. So that term is called as degenerate. So we spoke about this, right? So 4 power 3 is 64. So we have 64 codons and only 20 amino acids, which means more than 1 codon can code for an amino acid. So that process is called degenerate. Yes. So third one, so your genetic code is written without any punctuation, right? Any full stop, nothing. So it is written in a contiguous fashion. So that is one point. Then the next one, your code is universal. So it is a standard code from bacteria to human with a few exceptions. So you and I, we all have the same genetic code. So there is no difference. Fine. So it is universal from bacteria to human. So if you have a codon U, it definitely codes for phenylalanine only. So phenylalanine is an amino acid. Okay. Then the next salient feature is you have a codon called AUG. So if you have seen it in the animation before, you can recollect. So AUG is the start codon. So whenever your mRNA is about to get translated, the first codon that your tRNA, your ribosomes will encounter is your AUG. Fine. And AUG is also the initiator codon, and it codes for the amino acid methionine. Fine. Okay. The last point, you have stop codons. You have 3 stop codons which will help in terminating the process of translation. So the first one is UAA, UAG and UGA. Fine. So these 3 are the stop codons. So now let me go back and show you the genetic code. So first thing, it has 64 codons and 3 are stop codons. So can you quickly find where the stop codons are? So you have a stop codon here, you have 2 stop codons here, right? The next point, it is written in a contiguous fashion. So there is no full stop, comma, punctuation, etc. Right? And the third point, What was the third point? It is degenerate. You can see. So you have 4 codons coding for leucine, 4 coding for valence. So it is degenerate. Okay. So it has stop codons. And where is AUG? It codes for methionine, and it is also the start codon. Right? So is this much clear? Fine. Okay. So now going forward, so we will stop with this for today, because too much of saturation is also not good for your brain. Okay. So what we will talk about in the next class, so the next class where we will be continuing genetics is on Friday. Okay, so what happens in Friday's class? We will talk about the process of translation. We have just spoken about the introduction to genetic code. As such, I didn't tell you how the process of translation occurs. So we look into the process of how translation is happening. Okay, then we'll see the experiments as to how and who discovered DNA first. So how did we conclude ourselves? How did we convince ourselves that DNA is a genetic material and it was not proteins or RNA? So we'll look into a series of experiments. So you again have players acting there. Okay, so that is what we will see. So that is about for the next class. So the next class is on Friday, sharp at 4 p.m. So join me, and I promise not to disappoint you again. So make sure you like, subscribe. So if you need my PDF of my slides, so download the eCareerPoint app, so you will find my slides there. Yes, so thank you for joining me on this session. So have a nice evening. So we'll meet you tomorrow. So tomorrow I'm going to talk about class 11. So I'll be talking about the cell again. So tomorrow we'll talk about the superhero of biology, that is the cell. Okay, so hope to see you tomorrow, and thanks for joining me. Good day, bye. Oh shit.